Hi, everyone. I'm Georgina Sampat, the Global Marketing Coordinator for Floralife, and welcome to our first Fresh Cut Flowers webinar. This is a monthly initiative. Every last Thursday of the month, we will be hosting a webinar that focuses on flowers, flower varieties, and care and handling. And our goal is to bring you useful and educational information in 30 minutes. So on your screen, you can see a table with dates and topics. These are the next Fresh Cut Flower webinars that we are going to organize this year. Definitely take note of the ones that you're interested in and put it in your calendar. Before I hand over to our host, Technical Support Representative Emma Bradford, I just wanted to let you know that if you have any questions during the webinar, please send them to us through the chat. It's on the right side of your screen. At the very end, we'll do a little question session, and Emma will try and answer all your questions. This presentation will also be recorded, and after this webinar and a few days later, we will send you the link. So, Emma, take it away.
First of all, hello. Thank you for joining us on the first webinar of the series. Today we are going to go back to basics, so to Botany 101. Now, for some of you, this will be brand new information, so hopefully I'll go slow enough to explain it all. But for others, it'll just be a trip down memory lane, so maybe just a refresher of what you already know. So just to give you a summary of what I'm going to go over today. Firstly, I'm going to very briefly point out the three main differences between plant cells and animal cells. Then I'll describe how the plant kingdom is divided up by plant type. And then I'll go over some basic plant structures and how they work and why it matters. Now, some of you may remember seeing this type of cell diagram from secondary school or from high school biology. Don't worry, I'm not going to ask you to identify all the organelles. I'm just going to highlight the three main differences between the two types of cell. Now, on the left, you can see it as a plant cell diagram. On the right is your animal cell. Now, plants have plant cell walls, and that's the green area around that plant cell. The cell walls are there to provide strength, structure, and support. The other difference between the two is the plant cells have one large central vacuole. Now, what that does is it collects all the water in the cell, and it acts like a water balloon, and it controls the pressure inside the cell. So when that vacuole is full of water, it pressures up against the cell wall, and that provides turgidity to that cell. So when you've got plants that are nice and hydrated, nice and strong, you can see them. As soon as it starts to lose any water or start to wilt, that vacuole will shrink and that pressure will go down. So then that's when you see visible signs of wilting in your cut flowers or in your plants. And then lastly, the big main difference between the two are the chloroplasts. And these are the green organelles that you can see, and that is where the photosynthesis occurs. Now, within the plant kingdom, there are 5 main different types of plants which are divided into the following groups. Now, going from the most primitive to the most complex, from the left to the right, your first group are your charophytes, which contains all your green algae. The second group are your bryophytes, which contain your mosses and your liverworts. Now, these two groups are what's called non-vascular plants. Now, that means that they don't have any veins. So they don't have the veins to move the food and water to different parts of the plant. Now, these plants, for that reason, tend to stay small, and they must remain in contact with moisture at all times because they don't uptake the water from roots, they absorb the moisture through their tissues. Now, the last 3 groups are the vascular plants. These do have veins. And the first in the group are your pterophytes. That group contains your ferns and your horsetails. The second group are your gymnosperms. That is your conifers, your cycads, all your cone-bearing plants. And then lastly are your angiosperms, or the flowering plants. Now, those are the plant group that we're most familiar with, especially in horticulture. And that group is further divided into dicots and monocots, which I will go over. So, like I mentioned on the last slide, flowering plants, angiosperms, are divided into dicots and monocots. So, why the separation? Well, dicots and monocots differ in how they grow and in how their vascular systems or veins are arranged. Firstly, why are they even called dicots and monocots, also known as dicotyledon and monocotyledons? Well, as the name suggests, it comes from a leaf-like structure called a cotyledon. A cotyledon is one of the first things to emerge on a seedling, and they look like leaves, but they are in fact the food source for the emerging seedling. If you look on the picture in the very middle, there are two pictures of seedlings. You've got dicots on the left and monocots on the right. Dicot seedlings have two cotyledons, and monocot seedlings only have one. So that's an easy way to identify them. And the cotyledons on these pictures are the very first two leaf-like structures that you see on the left on the dicot, those two, especially the bean on the bottom. That smooth textured leaf, that first one that comes up, is actually a cotyledon. And then the second set of leaves that you see, that's the very first true leaves that you can see emerging. So that's an easy way to identify them. But the most important difference between the two are how they grow. Now, the area of growth on any plant is called the meristem. Now, this is just a sciencey name for the place where new cells are made, or area of growth, if you will. Now, on dicots, the meristem, or area of new growth, is located on the tips of the stems, whereas on monocots, on the other hand, the area of new growth is all along the stem. So if you look at the picture on the very right of the monocots, that orangey red area on the stem, that's the area of growth. And there'll be areas of growth like that all up and down that stem, everywhere there is a node. So what does that mean in real life? Well, if you cut the top of a tree off, and trees are dicots, it won't grow any taller from that point. Instead, it will just grow new shoots from the bottom and become more bushy. But when you cut your lawn, which is a monocot, you are cutting the tops of the plant off, but as the growing points are all along the stem, the grass will continue to grow taller and will need cutting regularly in order to stay trimmed. Now, this is also why tulips, which are also monocot, continue to grow after harvest, unless treated with Tulip 100, which prevents the growth of the stem. And that's also why, if you carve your initials into the base of a tree, which is a dicot, they will remain at the same height, no matter how tall that tree gets, because the growth occurs at the top of the tree only. Right, vascular bundles. So, first of all, what are they? Well, vascular bundles are the equivalent to arteries in humans. They're the main way that plants move water and food around the plant. So, although the vascular bundles in dicots and monocots do exactly the same thing, the way that they are arranged is different. Now, a vascular bundle consists of veins which carry water, which are known as the xylem, and veins which carry sugar, which are known as the phloem. Now, these are straw-like structures, and contrary to popular belief, cutting them at the stems at an angle will not increase the surface area of the vascular bundles. There's only so many that are there. It will, however, make it easier for you to push the stems into floral foam, so that angle will only help that. It won't increase the surface area. Now, as you can see on the left, in dicots, the vascular bundles are arranged in a ring around the stem, and in monocots, they are scattered throughout. Now, because rules are meant to be broken, some monocots have a solid stem all the way through, just like tulips. If you cut a tulip in half, you cut the stem in half, it'll be solid all the way through. but others are hollow in the middle. So like grasses, amaryllis, lilies, calla lilies, if you cut them in half, you will see that the inside of the stem is hollow. Now, if you look at the sunflower stem, which is a picture on the bottom left, which is a dicot, you can see that the center is actually white. Now, this white part is the non-vascular tissue, meaning that there are no veins in that part. The veins, or the vascular bundles, are all around the stem, in between the green skin and the white center. Now, because trees are also dicot, it is very easy to kill them, if you're that way inclined, by a process called girdling. Now, that is what has been done to the tree in the picture above. So by removing a section just 3 to 5 centimeters deep, all around the trunk, they have removed all of the vascular bundles from that tree section. So no more water or food can travel up the trunk. The center of the tree is just wood, which does not contain any vascular bundles. So there's no water carrying capacity within the inside of that tree. Right. So now you know a bit about vascular bundles and where they are. How do they actually work? Right. Well, sit tight, because the first time I learned what I'm about to tell you, my mind was blown. So how does water move through a plant? Well, simply put, it enters the root, moves up the xylem to the leaves, where it evaporates through the pores or stomata. Now, the sciencey word for this process is transpiration, and that's when the water evaporates through the pores or stomata. So far, so simple. How does it actually happen? Well, water molecules are actually very sticky, and they like to stick to each other, sort of like best friends holding hands. Now, all along the xylem veins, the water molecule form one long chain, each molecule holding hands with the next. So they form one long continuous chain of water from the roots all the way up to the leaves. Now, when one water molecule evaporates or is transpired through the pores on the leaf, this causes the whole water chain to move up by one molecule, which in turn causes another water molecule to be pulled into the roots from the soil. So the water is always moving from the roots up to the leaves in that way. Now, that process is why we recommend to only remove the leaves of cut flowers that will be below the waterline, as the remaining upper leaves will continue to help move the water up the stem by the same process. Now, the phloem, or sugar, on the other hand, can move up and down the plant depending on where it's needed. So in the spring, for instance, the sugar moves from the roots, where it has been stored over winter, up to the new shoots and leaves. But in the autumn, it will actually do the reverse and move down the plant, away from the leaves, to the roots, so the sugar can be stored for overwinter, ready for the next spring. The sugar in the plant will always move from wherever it's made or stored to wherever it's needed. All right. Now, with all that in mind, xylem vessels in cut flowers can become physically blocked by air bubbles, bacteria, or plant debris. And these blockages can prevent water from moving up the stem. So that's why we recommend to cut the stems to remove the blockages before placing them in water. However, Floralife have developed the Express technology range of post-harvest products to eliminate the need for recutting. So Floralife Express technology is designed to be used on flowers without having to recut. It does this by overcoming any blockages that may be present in the stem. So the blockages will still be there, but the Express solution will be able to move up the stem regardless. Right. So now you know a little bit more about stems and leaves. Let's finish with the flowers. So what you see here is a very simplified drawing of a flower. You will almost never see this drawing exactly represented in nature, but the sequence of where each part is formed will rarely vary. Now, there are 3 genes involved in the development of a flower: the A, the B, and the C genes. And depending on when they are turned on, will dictate which flower part is made. Now, the diagram on the top left shows a side view of a flower, and on the right, you can see an overhead view of the whorls or rings within that flower. Below is a depiction of the genes. Now, when the A gene is the first to turn on, and when it's on by itself, it forms the sepals, so the outer whorl. The sepals are usually green leaf-like outer parts of the flower, and when the flowers are in bud, the sepals are there to protect the petals. Then the B gene turns on while the A gene is still going, and when these two overlap, the petals are formed. Then the A gene turns off, the B gene continues, and the C gene gets turned on, and when the B and the C genes overlapped, that's when the stamens are formed. Now, the stamens are the male part of the flower and are made of 2 parts: the filament, which is the thin stem-like structure, and the anther, which is attached to the top of the filament, and then the anther contains the pollen. Then the B gene turns off, and the C gene continues. And when the C gene is on its own, the carpel or pistil is formed. Now, the carpel, or the other name for it is pistil, they're interchangeable, are the female part of the flower. And it consists of the ovary at the base, the style, which is a thin stem in the top, and then the stigma at the very top of the carpel, and which contains a sticky substance. So what I would really like you to take away from this slide, if nothing else, is the order in which the parts are formed. So working from the outside in, the parts are the sepals, the petals, the stamens, and the carpel. So what you are looking at here is a strawberry flower. And this is a pretty close representation to the previous diagram. So you can see the green sepals on the outside. Next row are the white petals, and then a ring of stamens and many carpels in the middle instead of just one. Now, if you picture a strawberry fruit, it has many seeds on the outside. And each one of those seeds is produced by one carpel. That's how many carpels are in the center of that flower. So there are 2 main forms in which you see flowers: single flowers and inflorescences. Now, single flowers are just what it sounds like, a single flower on a stem. An inflorescence, on the other hand, is simply a stem with multiple flowers on it. Like the example of a single rose and a spray rose, other examples include a standard carnation versus a spray carnation, or a standard chrysanthemum versus a spray chrysanthemum. And then other examples of flowers which come in inflorescences are delphiniums, agapanthus, veronica. There's many. So why does it matter if it's a single flower or an inflorescence? Well, it's because the flowers on inflorescence will be at different maturity levels. So imagine the flowers on the bottom of the stem or inflorescence are the firstborn. Then all the flowers produced after that are the younger siblings until you get to the baby at the very top. That's why the lower flowers will fade before the upper ones, because they're simply more mature. So as I said before, rules are meant to be broken, and you will encounter many flower oddities. So starting with the picture on the lower left, which is of a trillium, this flower is actually relatively normal and straightforward and is pretty close to the diagram that we saw first off. You can identify the green sepals on the outside, white petals, you can see a ring of stamens, and a carpel in the middle. So far, so good. Moving up to the tulip, however, you can see what looks like petals, stamens, and a carpel, but where are the sepals? Well, there are some bulb flowers which are special cases, one of the oddities. They don't have separate petals and sepals, but they have a petal structure which we call a tepal, and that's the case with the tulips. Now moving up to the calla lily and the anthurium, these are totally different beasts. Now I know what you're thinking. They look like they have one large petal and one central carpel, right? Wrong. Now what looks like a petal is actually a showy bract or a modified leaf. And what looks like a carpel in the middle is actually an inflorescence. So the sciencey terms for these are spathe and spadix. The spathe is the showy bract and the spadix is the inflorescence with lots of individual flowers. Now each red circle on the diagram in the center of the spadix represents one flower. So that green diagram with the red circles all around it, that is a representation of what the spadix is. And each one of those red circles is one flower. Now, why does that even matter, I hear you ask? Well, what may look like a petal isn't always so, and different structures behave differently. Now, because the spathe is a showy bract or modified leaf, it will behave more like leaf than it will a petal. So looking at some Dicot morphology now, if you look at the images on the left, they all look like pretty normal single flowers, right? Yeah, not so. So starting with the hydrangea flower on the very bottom left, again, what looks like a pink petal is actually a showy bract. So similar to the calla lily and the anthurium, again, why does it matter? Well, the structure of a bract is more like a leaf than a petal, right? And remembering that leaves have pores or stomata through which water transpires, this means that bracts lose more water than petals. And this is why hydrangeas are so thirsty, because they have more surface area to lose water through. And then moving up to the rose in the center, we see lots of petals around the outside and what looks like sepals where the stamens should be. So what's going on? Well, this is due to an error in the ABC gene sequence. So obviously a mutation has occurred which caused the A and the B gene to turn on together, and then the B gene stopped and just the A gene continued. So you have your A and B gene on together to make the petals, and then the A gene on its own to make the sepals. Now, the C gene is either absent or it doesn't work. It wasn't turned on at all in this case. And then lastly, saving the best for last, hold on to your seats because this is going to rock your world. So what you think is a single sunflower is not that at all. That single sunflower head is actually a flattened inflorescence. That's right. That sunflower is actually many individual flowers. So allow me to explain. If you look at the diagram on the right, you can see a cross section of the sunflower. Now, what looks like petals are actually ray flowers. So there's what looks like yellow petals all around the outside of that sunflower. Each petal is actually an individual flower. And those particular flowers are called ray flowers. And they only develop around the outside. All of the center flowers are called disc flowers. Now, if you picture a sunflower head full of seeds, each seed was produced by one single disc flower. And a disc flower is a very small flower which consists of a ring of fused petals with stamens and a central carpel. And this is true for all daisy flowers. So chrysanthemums, gerbera, asters, echinacea, dahlia, and even lettuce all have heads which are a cluster of smaller flowers. So, why do angiosperms even produce flowers? Well, to reproduce, right? But how does it happen? Well, to put it bluntly, the pollen needs to come into contact with the stigma and fertilize an ovule to produce a seed. That's basically what needs to happen. Now, some plants are very happy to self-fertilize, but some need the pollen from other plants, and this is called cross-pollination. And this tends to produce stronger offspring by avoiding inbreeding. Now, that said, how do plants encourage cross-pollination? Well, mostly by using animals. So, by providing a reward, usually in the form of nectar or by sacrificing some pollen, they can entice animals to visit their flowers. The animals then become coated with pollen and pass it on to the next flower they visit. Now, different plants use different strategies to entice mammals and birds. Others speak to bees. So the two center photos show flowers and how they look to us, so those are the yellow parts, and how they look to bees. Those are the parts on the top one on the right-hand side and the bottom one on the left-hand side. And what they've done is they've illuminated these flowers with ultraviolet light, so you can see the patterns that are revealed under the yellow. Now, these markings act as a landing strip to signal bees where to find nectar or pollen. And then lastly, if you look at the picture on the very bottom right, some plants don't need animals at all. They use the wind to move their pollen for them. And because they don't need to attract any animals, they don't produce any petals or nectar. And instead, they put all their energy into producing lots and lots of pollen that gets carried by the wind. That's why many people suffer from hay fever early in the spring when all the trees are flowering, because there's so much pollen in the air. Now, that's it for me. Hopefully you've learned something useful, or at least some fun facts to impress your friends with. So hopefully you enjoyed that all, and I'm happy to answer any questions.
Thank you, Emma. During your presentation, we got a few questions. So the next few minutes, we'll spend time on answering those. In the meantime, for our attendees, now and then a little poll will pop up. We'd love to hear from you, so feel free to answer any questions that we pose you. Emma, question number 1. What makes a sunflower follow the sun?
So the process is called heliotropism. And it's the same process that just about all plants do to track the sun. They move towards the light in any case. And technically, there's hormones involved which make one side grow longer than the other. But anyway, the reason sunflowers do it is because those bracts that enclose the flower head, those green bracts, in order to maximize photosynthesis, they've actually evolved to track the sun. So they do that on purpose, and they only do it when they're younger. Once the cells mature, they actually stay in a fixed position. So you'd often see sunflower fields that just all look one way at the sun. But when they're younger, they will actually track it from sunrise to sundown. And it's to maximize the amount of photosynthesis, basically.
Interesting. Let's see, the next question. Removing leaves below the water level is important. Should we also remove thorns below the water level?
Well, I don't recommend removing thorns at all. I know they're a pain because they're just a pain. But you want to avoid wounding your stems as much as possible. So plants are just like us. if you wound them, then there's an entry place for bacteria or infection, and the wound is going to cause a stress, just like it would for us. So you really want to keep your stems as intact as possible. The reason you want to remove your leaves below the waterline is that there'll be bacteria on the leaves which will be introduced to the water, and also when they start to decay, that also becomes food for any bacteria that's there. So you want to keep the stems below as clean as possible, but you also want to keep them intact, so don't remove any thorns anywhere.
Okay. Then we have, would tropical plants be different as to movement of xylem and phloem?
No. Yeah, no, it's still exactly the same. Whether they're monocots or dicots, a lot of tropicals are monocots, so things like ginger, strelitzia, things like that, a lot of them are actually monocots, but the movement of xylem and phloem will be exactly the same. The only thing that's different between them is how those vascular bundles are arranged within the stem, and then that meristem, that growth region.
Okay. We have a romantic asking a question. I buy pink roses regularly. I notice that not all varieties last as long as others. Am I doing something wrong?
Not at all. So if you imagine roses, they come in all different colors, right? Each different color will be a different variety. Now, what that means is, if you imagine, it's similar to dog breeds. Dogs are all the same species, but they come in lots of different breeds. And different breeds are good for different things, or they're more susceptible to different things, or they're stronger than others. They all vary. And it's exactly the same with flowers. So with roses, they're all the same species, but each different color will be an individual variety. So it's almost like an individual dog breed. And so the different breeds, different varieties will behave differently. So you really have to know your varieties and stick with the ones that you know will last, because some of them are just not bred for vase life. They're bred for yield or for color or for occasions, rather than sitting on your, nanny's shelf for a week and looking nice.
Thank you. Those were all our questions. So thank you, Emma, for your time, and thank you to all our attendees for attending. Definitely look out for the recording link. It should be in your mailbox tomorrow, and we'll see you at the next Fresh Cut Flowers webinar. Have a great day. Bye.
Bye.